This is part 2 of lecture 11, addressing what is a pathogen and understanding infection. In part 1, we talked about biological associations, parasitism, microbes as disease-causing agents, and now we're going to move into discussing microbial mechanisms of disease and think about, what virulence or pathogenicity factors actually are. And then we're going to conclude by describing the different stages of a microbial or infectious disease. First, let's define virulence as a measure of pathogenicity. Virulence encompasses factors that allow pathogens to overcome host defense or host defenses and multiply, thereby causing damage. Factors that are virulence factors are both defensive and offensive. Defensive virulence factors allow the microbe to survive in the host. Just remember that the host is actively trying to get rid of the microbe. Offensive virulence factors allow the microbe to actually harm the host. Let's consider some examples of both of these now. Defensive strategies used by microbes to survive in a host can include adhesions, capsules, and a variety of miscellaneous structures. Adhesions are cell surface molecules which help pathogens to attach to the host. For example, pili, hooks, spikes, and suction cups. An example of a pathogen that utilizes adhesions is Neisseria gonorrhoeae. It causes gonorrhea. And this organism binds to the membranes that are found in our urogenital tract. So I mentioned the urethra before, that's part of our urogenital tract. By attaching to the membranes of the urogenital tract, Neisseria gonorrhoeae is able to resist the flow of urine, which would otherwise flush the bacterium out of the urogenital tract. Let's look at some pictures and think a little bit more about adhesions. Adhesions can look different in different types of pathogens. C, D, and E are showing us images of a viral pathogen, of 2 bacterial pathogens, and of a eukaryotic pathogen that belongs to the group called protozoa, respectively. On the top, we have the human immunodeficiency virus, or HIV. It uses a category of adhesins called spikes. A lot of viruses use spikes. And those protein spikes are allowing it to recognize the T cells found in the human immune system. That's what HIV infects. The spikes allow the virus to specifically latch onto and infect those human T cells. In part D, we see an image of Salmonella bacterium and Treponema pallidum bacterium. Salmonella includes several species that are common causative agents of foodborne illness, and Salmonella use flagella both to locomote around and also as adhesins, allowing them to attach to the epithelial cells lining the human gastrointestinal tract. Treponema pallidum is the causative agent of syphilis, a very common STD. And Treponema pallidum uses a hook mechanism to attach to the epithelial cells lining the reproductive tracts or found in the reproductive tracts of both men and women. In both cases, these bacteria are using protein-rich structures in order to attach to host cells and thereby infect the host. On the bottom, we have Giardia. Giardia uses very large structures that are actually called suction discs. Giardia uses these suction discs to attach to human gastrointestinal tracts, the epithelial cells lining our gastrointestinal tracts. We get Giardia usually by drinking contaminated water, and those suction discs allow this protozoa to firmly attach to our GI tract so that it's not removed with the normal passages of food and nutrients through the GI tract. Very, very difficult to get the Giardia off your gastrointestinal tract lining once it attaches via suction discs. Other important defensive pathogenicity or virulence factors are capsules. And shown here are 2 images. On the right, we have Bacillus anthracis, and on the left, we have a bacterium called Streptococcus pneumoniae. These images are taken using two different types of microscopy. On the left, it is a routine light microscopy, and on the right, it's a special fluorescent light-based type of microscopy. Both of these organisms have capsules, which are extracellular matrices made primarily of carbohydrates, although in the case of Bacillus anthracis, this also includes a few specialized amino acids. The capsules allow these organisms to resist degradation or attack by the human immune system, and so they serve as defensive pathogenicity factors. There are a variety of other what we could call miscellaneous defensive pathogenicity factors, and an example is in Mycobacterium tuberculosis, or indeed any of the other Mycobacterium species that I've mentioned to you in class, for example, Mycobacterium leprae, which I mentioned in class the other day. This organism has a coat of mycolic acid, which makes up its cell wall. So there's no peptidoglycan, as I've told you before, in these organisms. Mycolic acid is very, very similar to paraffin wax. And so it's an extremely tough coating, and it helps, it's very, very effective at allowing Mycobacterium tuberculosis to survive inside some of the cells of our human immune system, which are called the phagocytes, for example, macrophage. And normally, bacteria would be degraded or broken down once they had been eaten by a human macrophage. But these cells of Mycobacterium tuberculosis, shown here, 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 and over here, okay, so all of this is a human macrophage. And the orange bits, okay, those are the M. tuberculosis. And it's surviving in there. It's doing just fine instead of being killed like it should be. And that's because it has this coating of mycolic acid, which is serving as a defensive pathogenicity factor. That's the nucleus right there, in case you were wondering. All right, so we looked at a few examples of defensive pathogenicity factors. Let's look at a few examples of offensive pathogenicity factors now. Let's consider exoenzymes and a variety of types of toxins, including both exo and endotoxins. Remember here that the prefix exo means outside or away from, whereas the prefix endo means inside or within. Exoenzymes are protein enzymes that are secreted by pathogens that serve to damage host or human tissue, thereby helping the pathogen to invade and multiply. And examples are collagenase, hemolysin, and leukocidin. Collagenase degrades connective tissue. Which is built primarily of the protein collagen. So in degrading connective tissue, a microbe can better invade a host or a human body. Hemolysin degrades or breaks down red blood cells. So a lot of blood-borne pathogens make hemolysin. Leukocidins degrade or break down white blood cells. In other words, cells of the human immune system, thereby helping to make sure that the host is damaged and the microbe thrives. A lot of different pathogens can make one, two, or all three of these type of exoenzymes. Exotoxins are proteins that are made within the pathogen that are released into host tissue. So they're made by the pathogen and then they're secreted out of the pathogen and they go into the host tissue. And there are a few different kinds of exotoxins. There are neurotoxins, cytotoxins, and enterotoxins. Neurotoxins interfere with the transmission of host nerve impulses, for example, tetanus toxin and botulinum toxin. In the next slide, we'll look at these again. Cytotoxins kill or damage host cells. It's a very general or generic term. And an example of a cytotoxin is pertussis toxin, which is secreted by the organism Bordetella pertussis, Bordetella, pertussis, as shown there. And that is the organism that causes the disease called, better known as whooping cough. Enterotoxins are a kind of exotoxin secreted into the host intestines. So this is very specific to pathogens that cause foodborne or waterborne illness. And they kill or damage our gastrointestinal cells. An example of an enterotoxin is cholera toxin, which is made by the organism Vibrio cholerae, which I spelled for you on an earlier slide. Let's think a little bit about two different kinds of neurotoxin. Tetanus toxin on the left and botulinum toxin on the right. So tetanus toxin is made by Clostridium tetani. And botulinum toxin is made by Clostridium botulinum. And they're both neurotoxins, and they're both very deadly, but they act quite differently. Tetanus toxin blocks the relaxation pathway, and it leaves the contraction pathway open. So when you've got tetanus toxin in your system, you remain permanently contracted. Every muscle in your body is contracted. In contrast, botulinum toxin blocks the contraction pathway. So you have what is called flaccid paralysis when you're exposed to botulinum toxin. So you can't move your muscles, you can't contract them at all. Whereas when you have tetanus toxin, you have what's called spastic paralysis, and that means that you can't relax your muscles. Both of these can be quite deadly, and they can result in fatality. Endotoxins, remember that the prefix endo means within. An endotoxin means that you have a structural component, typically the outer membrane of gram-negative pathogens, and it has toxic properties. It is the lipopolysaccharide outer membrane component that acts as an endotoxin, and it is released when the pathogen is dead. So you don't actually need to have live bacterial infection in order to see its effects. So you need to be exposed to the bacterium, and it's actually having parts of the bacterium within you that you are reacting to. Typical symptoms of poisoning by many gram-negatives include fever and strong immune response. A lot of different gram-negative pathogens cause that, so those are characteristics that indicate you've been exposed to endotoxins. So we've just discussed some of the qualities about pathogens that make them pathogenic, in other words, able to cause infectious disease in a host. And those qualities are known as virulence or pathogenicity factors, which is some aspect of a microbe's structure or physiology that allows it to be pathogenic. Let's think now about the stages of microbial or infectious disease. And the 5 classical stages are incubation, the prodromal period, illness, decline, and convalescence. Incubation is the period between initial infection, when you first catch the pathogen or when the level of the pathogen first rises inappropriately on or within your body, and appearance of symptoms. Remember that infection can be described as the presence of microbes in or on the body with indeterminate symptoms. The prodromal period is the period in which early symptoms appear, and it is not always well characterized. Illness is the period during which the disease is most acute and is accompanied by very characteristic symptoms. The decline period is the period during which the symptoms gradually subside, so you're starting to feel better, but you still have some symptoms. And the convalescence is the period during which symptoms disappear and recovery ensues. Let's look at this graphically. All right, so up the top axis we have number of microorganisms that would be within the body of the infected host over the course of illness, including the stages that I just described. And time is running across the bottom axis. So if we start over on the left, we begin with the incubation period. So you are exposed to the microbe, and then there is a short period of time, or a long period of time, depending on the pathogen, known as the incubation period, where you're not really seeing any symptoms yet, but the microbe is establishing itself within your body and beginning to grow. And here I want to remind you of the discussion that we had in class the other day about the microbial growth curve. I'm going to draw it up here. A microbial growth curve, that's the graph, it looks like this. Flat, up, flat, down, right? And that period right there is the lag period. That's the point in time where microbes are replicating their cell contents, cell numbers not yet increasing, but they are preparing to go through cell division. This right here is exponential or log growth, followed by that plateau, which is called stationary, and then the drop is the decline phase. You've got notes on all of that. So the incubation period would encompass the lag and perhaps early log phases of microbial growth. The prodromal period is where you're starting to see symptoms, and that's where you're really moving into exponential growth. And then you have a period of illness. And during the period of illness, you've got full-on symptoms, whether they're severe or whether they're, relatively mild. That just depends on the pathogen. And I would say that during that period of time, microbial growth is proceeding and you're really moving through exponential or log phase growth. Then at some point in time, your immune system will start to fight back, or perhaps the treatment that your doctor has prescribed will start to work. And at that point in time, we're moving into stationary period on the growth curve. The number of microbes is leveling out and actually starting to decline it. So you're in stationary into, early decline there. And you should see symptoms starting to go down. And then the period of convalescence would definitely be at the tail end of the growth curve, where your body is successfully fighting off all of these microbes and reducing their numbers either by completely clearing them out of the body in the case of some pathogens, or at least knocking them back down to the normal amount there found that allows you to have more of a mutualistic relationship with this pathogen. All right, and with that I'm going to conclude this part of the lecture. Please note that I have given you an accompanying question sheet that I would like you to use as you take notes on these lectures, and I'd like to check that when I see you next in class.